long day. Made a mistake. I should not have gone to grab some food. I should have studied as soon as I got off work. How many cards do I have? 224. Okay, well, it's been worse. I knocked down a whole bunch this morning. We've got time. All right. This is a team sport, guys. It's you and me. We're gonna make this work. Contraction. Okay, cool. So, hypertonic contraction. Heavy sweating, loss of excess water without equivalent loss of sodium chloride. Both ECF and ICF volumes decrease. Right. Isotonic contraction. All right. So these are easy cards. Hypotonic contraction. Loss of salt. Okay, so it's important to understand that when we're talking about the tonicity, we're talking about what's going on. So we lose sodium when it's going hypotonic. What can we expect to happen? Well, the extracellular fluid volume is going to decrease, right? And so therefore, cells are going to swell. So the body fluid change is going to be that the ECF goes down, the ICF goes up. Same amount of water, by the way. Fantastic. Okay, cool. So, let's see, and there's, okay, look, it's season 2 of The Tick. Oh, that's fun. Ah, man, I'm getting so distracted. All these good shows are coming out. We're gonna get HBO. I gotta hydrate, too. Let's see. Sorry, guys. All right, all 0 viewers. Hello, okay. So, let's see, intracellular, okay, cool. So what are my 3 categories? G protein coupled receptors, inotropic. What is my other receptor type? Somebody remind me. Shoot. Catalytic, right? Catalytic, enzyme linked. Yes, bam, 25 minutes. Cool, all right, what else do we have here? Lots of them. All right, so muscarinic peptide, adrenergic. What am I missing? Hormone. No. So GPCR is on the cell surface. Inotropic, no, that's not the same thing. Diversity of GPCR. Adrenergic, muscarinic, which is cholinergic receptor, peptide, glycoprotein. What am I missing? Hormone? Steroid receptors. No, those are intracellular and those are catalytic receptors. Not sure. Let's see if we can find some hints down here. Nope, not going to. All right, calcium receptors. Right, calcium receptors. Of course, calcium receptors. I'd love an example of what a calcium receptor does. That would be helpful. Wait, undo. Yeah, 1 minute. Jeez. It's a new card. Okay, stimulates, what do we got? Erythropoietin, excreted by the kidneys. 4.5, that's 4.4 in the plasma for potassium, or milliequivalents per liter. Values for normal, lean, 70 kilogram man. What do we got? We have total body water, blah, 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 blah. Okay. 4 to 5%, plasma volume is 4 to 5% of the body weight. Okay, reflection coefficient permeability, urea, goes into the cells, causes cells to swell. Notion to cell osmolarity because the water follows it. Capillary wall. Okay, so this is asking about, so water is forced out into the interstitium. Filtration. All right, sodium chloride is confined to the ECF. Rise, fall are same. What happens to the osmotic potential? Osmotic equilibrium. Wait, no, what happens to the ICF? Okay, cool. So for hypertonic contraction, or hypertonic expansion, that means that I've increased my solute volume. Therefore, I'm going to pull water out of the cells and into the extracellular space. Right, decrease in ICF, increase in ECF. Pretty easy. Okay, great. The last one I was forgetting was the calcium receptor. So calcium, anything else in there? CARs. I don't know anything about these guys. I have to look them up later. All right, number 4. Okay, so the first 3 steps. So ligand binding to the protein binding to GCF. Right. So GEF. So next we're going to have the alpha and beta are going to move and bind to the other thing. G protein dissociates from the receptor. Okay, great. So alpha and beta subunits bind. Now they can interact with the appropriate effectors E1 and E2. So, right, that would make sense. So I'm going to turn that feature off. Let's see, extracellular fluid. Oh, right. So extracellular fluid. So that'll be the remaining 33% of total body water. That's pretty easy. Let's see, GPCR activation effector molecules pathway number 4. So GPCR beta gamma. Hello, welcome viewer. Hi, how do you do? Don't forget to follow. Let's see, GPCR beta gamma subunits. All right, so for the GPCR, the beta gamma subunits were blank, i.e., they attach to the G protein in the plasma membrane. Membrane bound? No. So this is the 4th iteration. Beta gamma subunits. I mean, originally described as simply anchor proteins. Okay, how much do I care about remembering that? I might not really. So we're going to bury that card. We're going to end up unburying a lot of these guys anyway, because as soon as we get to the end of this deck, we're going to need to unbury our cards so that we get the other cards that are in each of these zones that we've already viewed. So because they attach to the plasma membrane, which is good to remember, because if we're looking at the illustration, then they do not look. It does not look like the beta gamma subunits attached to the G protein to the plasma membrane. It doesn't look like that. It looks, because they do duck beneath. They do when they bind to GTP, they seem to move around a lot. The alpha subunit interacts with the cyclase, or whatever it is, the transfer protein that helps the conformation change to allow the DP to be released and the GTP to attach. Then after that they dissociate, and then they go and they combine with the E1 and E2 proteins, or I guess that's probably a factor 1 and factor 2. So it doesn't seem like they attach unless they're sliding around in the membrane. Maybe they must not be on a lipid raft. I'll have to look up more about that. Yeah, this is the card that I was talking about. Okay, cool. So let's see. So ligand binding results in the binding of G protein to connect to the receptor and catalyze the GTP to GDP. GF, well, right, that's what I was trying to refer to. Guanine nucleotide exchange factor binds the alpha subunit, facilitating the dissociation of the binding of GTP. Yes. And then beta and gamma dissociate from the G protein, from the receptor, and the G protein, right. The G protein dissociates from the receptor. Great. And illustration here, ligand binds to the receptor activates, and then so you can see them clearly in the membrane. So I'm wrong. So that it does look like they are, and it looks like they slide around in the raft, or they slide around in the membrane. So they remain attached. They stay localized. At no point did they dissociate and then have to reassociate, from the membrane entirely. So there's not a lot of three-dimensional movement. So this restriction actually must make it a lot easier for them to find and interact with the E, because they have limited couple fewer degrees of freedom, because they can't rotate in around one axis either. You can't rotate around one axis. Actually, 2 axes. So they have 3 fewer degrees of freedom, and that makes entropy a lot less for them. Okay, so receptors and signaling 1. Summary of key concepts. Hormones elicit physiological changes. All right, these receptors, that's different in different tissues. These receptors occur at different locations depending on the type of the hormone. So it depends on the tissue, depends on the hormone. So the effect depends on the target. It also depends on the messenger. Both of those make sense. Receptors for lipid-insoluble peptides or catecholamine hormones are on the cell surface of the plasma membrane. Great. Receptors for blank are blank in the cytosol and nucleus. Receptors for steroids are free-floating in the cytosol. Receptors for lipid-insoluble... Oh, okay, so there's a whole list. So lipid-soluble steroids, thyroid hormone, or vitamin D. So I should have remembered these, and I probably would have been able to if I had indicated to myself that I needed a list of 3. So we're going to do that here. We're going to say list of 3, and this is how you format that. So now when I go down, I get a list of 3, right? Steroids, thyroid hormone, and shoot. Wow, okay, cool. Well, clearly I needed that. All right, so I can't remember from this morning, but I'm going to try. I can't remember from just a moment ago. Okay, thyroid hormone, vitamin D. Right, thyroid hormone, vitamin D. Seriously, vitamin D, yeah. Okay, general characteristics. right? So this is the calcium receptor, CR. That's one that's missing from here. I need to know more about what those do. Here we go. So there are 5 major classes of catalytic receptor, right? Guanylyl cyclase, the tyrosine, receptor tyrosine, tyrosine receptor, or tyrosine receptor tyrosine kinase. Then there's a tyrosine kinase associated receptor. Then there's the tyrosine phosphatase receptor complex. So that's 3, 4, I guess, guanylyl cyclase. And then there's one more that I'm forgetting. I don't remember. Oh, right, the serine threonine kinases. Rats. Okay, try it again in a moment. Okay, low sodium. I sure hope so. Receptor guanylyl cyclase, receptors. All right, so atrial natriuretic peptide. So that I should remember. What does that do, Noah? Well, great question. That actually senses the presence of how much fluid is in the extracellular space in the atrium. And when that becomes too much, then it assumes that that's because there's too much sodium in there, which is correct. And so then it causes the kidneys, secretes this hormone, which then causes the kidneys to reuptake's the wrong word, to recover whatever fewer sodium ions. Let's see. Receptor shown left. Upon ligand binding domain, receptors dimerize and intracellular transactivators? Dimerize and guanylyl cyclase joins. Right, well, that makes sense. All right, we're going to say yes to that. All right, receptors, list of 3. Okay, cool. So that was easy. Steroid, steroid hormone, thyroid hormone, and vitamin D3. Right, soluble steroids, lipid-soluble steroids. And the important part about this is that in order for them to go, receptors for these guys in the cytosolic nucleus, they have to be able to get through the membrane. These guys are not going through channels. They have to dissolve and go through the membrane. They might have carrier proteins. Now, they might have carrier proteins on either side, but I'm not sure. All right, GPCR, beta-gamma subunits, modulation of effector proteins. All right, GPCR, beta and gamma subunits were originally described as simply anchor proteins. They're right, because they attach to the plasma membrane. You attach the G protein to the plasma membrane. Now, I'm pretty sure that the G protein... Oh, right, the G protein, yes. So that must be the guanylyl cyclase. Is that the G protein that's referred to? Let's see. I'll look it up in a moment. Let's see. G protein, also named guanine nucleotide binding proteins. Right, okay, it's cool. So that would be the okay. So literally just beta gamma. This receptor binds the G proteins to the matrix. But the G proteins bind the receptor to the matrix. Okay. So 5 major classes. Serine threonine is one that I keep forgetting. Guanylyl cyclase, which is atrial natriuretic peptide. Serine threonine, I need to look up what some examples of those are, or I'm sure they're in a card somewhere. I need to remember them. Tyrosine, or receptor tyrosine kinase. That is the I'll have to remember some better examples of that. Receptor tyrosine phosphatase and tyrosine kinase associated receptor. Those are my 5. Yep. All right. Catalytic. Causing change and the conformation of the receptor, tetramerization, interaction of the beta gamma dimerize, and blank by blank of the blank. I don't even remember anymore. Insulin receptors. So insulin receptors are receptor tyrosine kinases. Okay, cool. So those are my So the nerve growth factor and insulin receptor. Those are my 2 primary examples of receptor tyrosine kinase. So I have a atrial natriuretic peptide for my guanylyl cyclase. I've got receptor tyrosine kinase, nerve growth factor and insulin receptor as my receptor. Nerve growth factor receptor and insulin receptor as my receptor tyrosine kinases. These are tetramers. Okay, I need to be able to call these things to mind when I think of it, because when I'm in my test, I'm going to want to know for sure. I want to have an explicit episodic memory. In both cases, the activated tyrosine kinase directs the tyrosine phosphorylation of various cytosolic proteins. Yes. Okay, cool. So now what is going on here? Conformation. Yeah, chain of conformation. Great. All beta chains. Okay, well, that's sensible. And activation by autophosphorylation of the intrinsic tyrosine kinases. Okay, change conformation of the alpha beta chains. Okay, the dimerize, then that's what activates them. That is fair. I'm giving myself 1 minute. I'm not sure. Call it again. All right, ligand binding, and then, but basically this step is just that the G proteins, the alpha, beta, gamma subunits, dissociate from the receptor. All right, consists of a, all right, so here we go. So tyrosine kinase associated receptors. Okay, so my receptor tyrosine kinases, those were associated with, okay, so those were nerve growth factor and insulin receptor. Nerve growth factor receptor and insulin receptor. For my tyrosine kinase associated receptor, I got growth hormone and interleukin 6. These are my two prototypes, my two examples. They consist of a receptor with an extracellular domain and alpha beta intracellular domain. Transmembrane intracellular. Well, that makes sense. So these can be contrasted with GPCRs because they have a single membrane, transmembrane domain. Single membrane molecule? What the heck? Whatever. There are two of them. The insulin receptor, causing change of conformation of the receptor, leading to the interaction of alpha and beta chains and activation by dimerization. By autophosphorylation of the intrinsic tyrosine kinase. Okay, autophosphorylate. Wait. The insulin receptor is a tetramer of two alpha and beta bind. Insulin binds to the alpha chain, causing chain conformation receptor, leading to the interaction of alpha and beta chains, the activation by autophosphorylation of the intrinsic Okay, so what you're saying is that the what activates? It's activated by the autophosphorylation. Okay. So it phosphorylates itself to then activate the tyrosine kinase. So then what tyrosine exactly does a kinase? I assume that, tyrosine is an amino acid. I assume it must be not some random tyrosine, but some tyrosine on the surface to which the receptor binds on some molecule, and it just goes around phosphorylating all the proteins that the receptor is specific for, and that's the second messenger. In both cases, the activated tyrosine kinase directs the tyrosine phosphorylation of various cytosolic proteins. Okay. Yes. Okay. Again, tyrosine phosphorylation of various cytosolic proteins. Right. So it chooses so it has an allosteric active site or whatever, a spot on the protein to phosphorylate and changes conformation to then make it an active protein. Or an inactive protein, who knows? So, yes, NGF receptor, and they look completely different. Alright, so let's see. Causing a change in the conformation of the receptor leading to the interaction of the alpha beta chains and activation by autophosphorylation of the intrinsic tyrosine kinase. Great. Example, neurotic peptide, right, so the guanylyl cyclase right away, I know that. Ligand binding to the receptor domains, receptor shown left, let's see, upon ligand binding to this domain, receptors blank, dimerize, intracellular tyrosine guanylyl cyclase domains are activated. Dimerize and guanylyl cyclase domains are activated. Great, wow, I got it right. Look at that. All right, receptors. List of theory. Steroid hormone receptors. So tired. Steroid hormone receptors, vitamin D3 receptors. Or no, receptors for steroid hormones, vitamin D3, and specifically lipid soluble steroids. And what's the other one? Thyroid hormone. This receptor tyrosine kinase tyrosine kinase associated receptor, and receptor tyrosine phosphatase, and serine threonine protease. Or, I mean, kinase, whatever. Let's see. Causing a change in the conformation of the receptor. Okay, so the insulin receptor, 2 alpha 2 beta. So insulin receptor is a so I've got two: nerve growth factor and insulin receptor. These are both tyrosine receptor tyrosine kinases. Yes, receptor tyrosine kinases. Again, nerve factor, insulin. Nerves need sugar to grow. Let's just remember that way. And they're very direct about it. So it's not the associated, it's the actual one. So let's pick a mnemonic. Causing change in the blank of the receptor, leading to interaction of alpha, conformation, interaction of alpha beta chains, and activation by autophosphorylation of the intrinsic receptor kinase, intrinsic tyrosine kinase. Yes. Hormone binding converts. Okay, so this is for intrinsic receptors. Hormone binding converts the receptor into transcription factor, which then initiators. No, initiator regions. Yes, activator regions. I don't know. Start codons. No. Origin recognition. No. AUG. No. Hormone response elements. That's a good vocab word. Oh, hormone response elements. It's amazing. I mean, I've read that a few times, and I just would not remember hormone response elements. This is why the flashcards are important, guys. This is why the flashcards are important. All right, plasma volume, clinically important. Subcompartment of ECF. We use 1 through 1 iodine labeled albumin, or that blue dye, what is it called? Evan's blue. Evan. All right, I know a guy named Evan. I should just remember his name. Gosh darn, look at that. It would have been 1.2 months. Nope, 10 minutes. All right, intracellular domains. Thyroid hormone receptor. In the case of THR, it is already present in the nucleus. Right. Oh, wait, we already know what this is, though, right? Right. It is, in fact, it is a hormone receptor, thyroid hormone, along with, what are the other two? Vitamin D3, and what else? Lipid-soluble steroids. Tyrosine associated kinase? No, they're intracellular receptors. These are the 3 intracellular receptors. And man, I should have read that bullet point better. That's what the card looks like. You gotta remember the context of it, otherwise you're not learning anything. So there's, it's a good thing that we're making connections. And the most important thing is make connections between the cards. Otherwise you're not gonna catch these things, and you're not gonna be really learning anything. Okay, blank enters a cell. The steroid, lipid-soluble steroid. Now, what are we looking at here? Thyroid hormone. Thyroid hormone enters the cell. Enters the nucleus, binds onto the repressor, and transcription, deactivating the repressor, and transcription is initiated. T3, THR, enters the nucleus, THR. Okay, good. Thyroid hormone receptor, which is also a repressor. Okay, and this is one I've gotten wrong. Growth hormone and interleukin-6 are tyrosine associated kinase. Yes. Okay. Cool. This guy. Growth hormone and interleukin-6 are. Why? Because you gotta grow, and interleukin-6, IL-6. What does that do? IL-6. I need a reminder. All right. Let's pull up Wikipedia over here. I'm gonna use my thing that I used to monitor you guys. Hello. Hello. How do I... somehow I've lost my exclamation mark. Oh, there it is. Hello. There we go. Okay, alright, so what do we care about? Interleukin-6. IL-6. IL-6. There we go. Illinois 6. No. Interleukin-6. Pro-inflammatory cytokine. Interleukin-6. Alright, and I'm going to post this on my screen over here. I'll pull it up for you guys. Right there. This is interleukin-6. And let's see, let's read about it. Okay, interleukin-6 is an interleukin that acts both as a pro-inflammatory cytokine and anti-inflammatory... What? An anti-inflammatory myokine? What the heck? What is a myokine? A myokine is one of several hundred cytokines or other small proteins and proteoglycan peptides that are produced and released by muscle cells, myocytes, in response to muscular contractions. They have autocrine, paracrine, and or endocrine effects. Their systemic effects occur at picomolar concentrations. Fantastic! This must be recently discovered. What are the citations on this page? I mean, I have never heard of this before. It seems like recent. If these are picomolar concentrations, I mean, I may be totally wrong, but it seems like this is something that's must be recent. No, let's see. The first article that's cited is one from 2007. Role of myokines in muscle and exercise. What are these other ones? Okay, here we go. But number 2 is 2018. Endocrine crosstalk between skeletal muscle and the brain. Frontiers in neurology. Fascinating. Muscle 2012. Okay, so this is kind of recent stuff. I had not heard of myokines, but they're old ones. Exercise induced increased serum interleukin 6. So it was discovered in, it seems like it was called a myokine, probably in, it must have been in 2007-ish, and it has since gotten more popular. It takes a long time. This is a fantastically well-researched Wikipedia article. I am consistently impressed by Wikipedia. I wish I could make better concentrations, but people have already contributed so much that you really have to be an expert in a specific thing in order. I mean, if it makes sense though, right? Because if all you research, if you have a year-long project, 3 years project working on interleukin 6, then you better bet that you're going to make a contribution. You are going to make a contribution to the Wikipedia page for IL-6. People spend so much time on it, it's ridiculous. In fact, I would jump at the chance. Once I work on something intensely enough, I will jump at the chance to, especially a molecular biologist, I will jump at the chance to write in a Wikipedia article about the thing that I'm interested in working on. And in fact, I might even be, another cool thing that you can do is if you're at a university and you attend lots of talks, you take the opportunity to start to add to Wikipedia articles when you're the first one to, learn, one of the first people to learn about something that hasn't quite been published yet, or is about to be published. You can go to the talk and whatever they say in the talk, you can then you might then be able to add it to the Wikipedia article as long as it's publishing inside it. I mean, I wouldn't do that with everything. Like, you shouldn't believe everything you hear every talk you listen to, but it wouldn't be a bad idea, Okay, growth hormone receptor. I wonder if I'm myself here. Let's see, IRL, description of the stream. In which we learn all about medical, I can't type, about space, medical space, physiology. Add stream marker. Well, whatever. Tags. Oh, here we go. Tags. Medicine. Nope. Educational. There we go. Yes, I am all about educating viewers. There we go. Educational. All right, now we're cooking with fire. Now when people search for educational, I didn't even know that was a tag. I'm gonna search for other educational Twitch streamers. We're gonna see. We're gonna see what else is out there. I mean, nothing will be as dry and boring as flashcards, but so I think I'll have one. I'll definitely be winning on dry and boring. Yes. All I ever wanted. Single membrane molecule. Single membrane molecule. I somehow feel like that's not right. Whatever. The steroid hormones, thyroid hormone and vitamin D3 metabolite. Right, okay, cool. Intracellular. So this is in the category again of intracellular receptors. I keep wanting to make these GPCRs, or I keep wanting to make these catalytic receptors, but they're not. We've got a few kinds. We've got GPCRs, catalytic receptors, inotropic receptors, and we have the intracellular receptors. And it's such a stupid name, but literally it's not functional at all. So it's not very functional. It's just they're inside of the cell, which basically means that it only refers to things that can at least partially dissolve in lipids, can make it through the membrane, and get into the cell. So, whatever. whatever. Oh, and the calcium receptors. Jeez, that's the other one. Unless it's no, actually I think calcium receptor. I think that's a subset of GPCRs. So my bad. Okay, man, we've been streaming for 30 minutes already. Can you believe it? V for reference. Oh, right, that's a really good Wikipedia thing. Should I purchase it? Ooh, should I purchase it? No, I don't have any money. Guys. Oh man, oh man. Man, oh man, oh man, oh man. Sorry, I'm getting distracted. Sorry, you guys. I just have to look something up real quick. Let's see. Find my friends. There we go. I'll see where my human is. Let's see. Is she on her way home yet? Yep, she is still at her defense class. Looks like it. Man, this go late. Probably ends at 9:30. So I guess 2 more minutes. Okay, retinic acid receptor. What's going on here? Okay, so it starts the DNA called receptor spots. No, wait. Shoot. They had a snazzy name. Hormone responsive elements. That's right. Hormone responsive elements. What do? They respond to hormone. What are they? They're elements. That's right, because elements are parts of the DNA. Causing change in conformation of the receptor. All right, so what am I talking about here? I'm talking about insulin. Insulin. And what is that? It's an intracellular receptor. No, it's not. No, it's not. It's a catalytic receptor. What kind of catalytic receptor, and what's it coupled with? Well, growing nerves requires sugar. Sure. Brain requires glucose. So nerve growth factor is another example of this. Now, how does it work? Let's see, is it tyrosine associated? Tyrosine kinase associated, or is it a receptor tyrosine kinase? I tend to think it's a receptor tyrosine kinase because it's very direct. The connection is very direct. And that's why mnemonics are so important. Okay. Causing a change in the conformation of the receptor, leading to interaction between alpha-beta chains of the, guys, humans. Let's see. I'll make a Gengar. I do like Gengars. We're just gonna do that. Alpha beta chains and blank by blank, and they dimerize. They activate by autophosphorylation of the tyrosine catalytic domain, or tyrosine kinase domain. Intrinsic tyrosine kinase, yes. Intrinsic meaning is part of it. Ionotropic and calcium, and these are cell surface receptors. Okay, G protein, ionotropic, and catalytic. These are all cell surface receptors. The 2 general categories, right? Again, 2 general categories, and those are the cell surface receptors and the intracellular receptors. All right, contraction again. Hypotonic means you lose sodium. That means the water goes into the cells, they swell, it goes out of the extracellular fluid. Blank enters the target cell, enters the nucleus, and binds onto the hormone response element. And this would be T3. And this would be the, not the hormone response elements, but it would be the thyroid hormone responsible element, the THR. Okay, that was Evans blue, and I know an Evan, that's how I remember it. Attached to plasma proteins. All right, retinoic acid receptor. All right, hormone binding, hormone response element. Yep, cool. Lost in desert, loses more salt than water. Decrease. It'll increase. It'll go down. It needs water. You want to put water directly into the cells and slowly leave. You give them dextrose. Or no, not dextrose. Yeah. Yeah. Don't want to lyse the cells because then the calcium will go everywhere. And if the calcium goes up, then the heart will go into atrial fibrillation. Yeah, actually, that's a great question. What happens in hyperkalemia? Heart Hyperkalemia. Ah, not the App Store. Jeez. Yeah, cardiac arrhythmias. Arrhythmia. That means no rhythm. Now, I was giving the benefit of the doubt that it could just kind of fibrillate a little bit, but apparently not. Wait, I thought arrhythmia would be, not dysrhythmia. Grr. Oh, no. Arrhythmia means no rhythm, not no heartbeat. Ah, yes. Yeah, you find somebody with a crushing injury and they're just gonna go crazy on you. Crazy. Okay, whatever. Yes, that's correct. Nuclei. Those are clusters of neurons. Presumably that's where the thinking happens. Ooh, thinking. Yay, groups of cell bodies, CNS. Alright, the steroid hormones. Blank and the vitamin D3 metabolite enter the target cell and bind to the receptors that are either in the cytoplasm or in the nucleus. Steroid hormone receptor binds to the DNA as dimers, as shown below. Yes, what are we talking about? Steroid hormones, thyroid hormone, and D3. Intracellular. Alright. Nervous system can also be divided into functional components, right? Somatic, autonomic, regulates heart rate, breathing. Right. Okay, diversity GPCR, calcium also, calcium receptor. Main parts of the central nervous system. Nerves are part of the central nervous system, bundles of axons, groups of neurons, and glia. Axons and glia, cell bodies. Ganglia, right? They're cell bodies and glia. Through division, sympathetic, autonomic, and parasympathetic, right? Parasympathetic. The divisions of autonomic. Yes, I am doing that. One sec. Alright, well, I'm going to pause with you guys for a moment. Later, Gator.